Now we are going to talk about very important topic in our lectures, which is the sterilization indicator. What is the meaning of sterilization indicators? Or in another word, how to monitor the sterilization process. How to monitor our autoclave. How to monitor our filters. How to monitor our radiation apparatus. How to monitor these apparatus. First, to test the efficacy of the autoclave, there are different methods for checking the efficacy or monitoring the efficacy of autoclave. The first is the mechanical method. Each time we use the autoclave, we must be sure that the autoclave and its lid is efficient. We must take a printout or graph that monitors the time, temperature, and pressure cycle. This is in the digital autoclaves, but in the ordinary or the old type autoclave, we must be sure that the pressure inside is double the atmospheric pressure from the pressure gauge that is present. We must monitor the autoclave machine, and so it is called mechanical method. We must be sure that the autoclave as a machine, all the part is act efficiently. This is a mechanical method, and as we said, in the modern or digital autoclaves, there is a printout of graphs that give the time and temperature and pressure cycle inside the autoclave. Chemical indicators. There are different methods for chemical indicators, but the principle of all the methods is here papers or even tubes containing a fluid, or papers, strips impregnated with a fluid. All these fluids make chemical reaction at the temperature and the pressure inside the autoclave. That means the bronze tube, for example, containing inside a fluid that make a chemical reaction at 121 degrees for 20 minutes, which are the conditions inside the autoclave. If the autoclave reach its temperature and pressure efficiently, there is a chemical reaction occur inside the bronze tube, and this chemical reaction will lead to color change. And after sterilization process, I will monitor this bronze tube. If the color change occur, that means the temperature and the pressure inside the autoclave reach the accurate temperature and pressure, and so the sterilization process is efficient. But if the color change is not occur, that means the temperature is not rise to 121 and the pressure not rise to double the atmospheric pressure, and so the sterilization process is not efficient. This is the principle of the chemical indicator method in all types, in bronze tube, in witness tube, in the paper strips. The principle is that these strips or tubes containing or impregnated with a chemical compound that make chemical reaction at the specific temperature and pressure inside the autoclave. And if it's occurred, it results in color change. And I can check this chemical indicator after the sterilization process by this color change. Finally, the biological method for monitoring the sterilization effect of the autoclave. Bio, that means alive. Biological method, that means I use a biological or alive organism. In case of autoclave, we use the spores of bacteria called Geobacillus stereothermophilus. Why this strain especially? Because this strain is very, very resistant to heat and can be alive to temperature near 120. It can still alive. And so, if we use these spores inside the sterilization process of autoclave, and after sterilization process, I will try to cultivate this organism. If the organism is cultivated, it will be not efficient sterilization process as the organism not killed inside the autoclave. And if the organism is not cultivated, this means that autoclave is efficient. Again, this is the biological indicator ampoules. The ampoule is provided with inside plastic tube that's containing the spores. It's containing culture media. The ampoule consists of two ampoules inside each other. The inside ampoule that contains this red fluid is the culture media. Culture media for cultivation of the organism. Where is the organism? It's present in the filter paper at the bottom of the plastic tube, filter paper impregnated with the spores. I have two plastic ampoules, the inside one containing the culture media responsible for cultivation of the Geobacillus thermophilus, and these spores is present impregnated inside the filter paper inside the at the bottom of the tubes. We add these ampoules inside the autoclave and make the sterilization cycle normally. If the sterilization cycle reaches to 121 degrees, the organism must be died. And if the autoclave not reached to 121 degrees, the organism will be still alive. Okay, how can I check this after the end of the sterilization cycle? I will make pressure in the ampoules, and the culture media will be around the filter paper containing the spores, and I put it inside the incubator. If the organism present, it will be cultivated, and it will make its metabolism and change the pH and give this yellow color. That means the organism is present and make its metabolism and change the color of the media, and so the autoclave not kill this organism, and so the autoclave is not efficient. But if the tube and the culture media inside the tube still red in color, that means the organism is died, and so the sterilization process is efficient. This is the principle of the biological method or biological indicator used for monitoring the sterilization efficacy in case of autoclave. The same principle can be used in the radiation, but the organism used will be different. The organism used in case of radiation, we use the organism that's very, very resistant to radiation, and so on. Okay, now how to test the efficacy of filters. How to test the efficacy of our filters. We prepare a suspension of organism that is characterized by its very, very small size. This bacteria is called Serratia marcescens. Serratia marcescens is a bacteria have very, very small size. And I prepare suspension of this Serratia marcescens and begin to filter it through my filter. After that, I will take the fluid after filtration and try to cultivate it in a suitable media. If there is a cultures of Serratia marcescens, it appear, that means the filter pass the Serratia marcescens, and so the filter is not efficient. So Serratia marcescens can be used in the monitoring of filter paper, again because it's very small in size, also because it gives red colored colonies upon cultivation, and so it will be very clear to be detected. This is a method to test the efficacy of filters. To test the efficacy of ionizing radiation, biological method, as we discussed before, but the organism here will be Bacillus pamilus. This Bacillus pamilus is very resistant spore to the radiation. Finally, we are going to talk about the chemical methods of sterilization. And as we said, most of the chemical methods of sterilization are disinfectant and used mainly for decontamination of the object and freeing it from the most pathogenic microorganisms. This object may be non living object, as we said before, or maybe living tissue. It's called antiseptics. There are liquids and gases. These chemical disinfectants are called the non-specific antimicrobial agents, or called non-antibiotics antimicrobial agents, including liquid and gases. We'll take it in the short form, only the example and its mechanism of action on bacterial cell. First, ethyl alcohol 70%. Ethyl alcohol 70%. It makes denaturation of the protein and dissolve the lipids of the bacterial cell wall, and so it's used as a disinfectant. Disinfectant for thermometer in the hospital can be used as a disinfectant for thermometer. Also, it can be used safely on the skin, wound, and even the normal skin before injection. Okay, so it's called antiseptic. Antiseptic. Ethyl alcohol 70%. Okay, can we use ethyl alcohol 100% to have much more better effect? Why 70%? No, 70% is calculated specifically, and this part of water, 30% water, is necessary to provide sufficient polarity of the solution, and so the solution can enter inside the bacterial cell and give its effect. Okay, it is the first liquid we study, ethyl alcohol 70%, as a form of antiseptics. The second, the phenolics or the phenols. It causes disruption of the plasma membrane of the bacteria. And shortly, you should know that it can be used for decontamination and disinfection of the floors in 5% concentration. And in case of the vaccine, and it can be used as a preservative for vaccine in 0.5% concentration. Halogens make oxidation of the proteins of bacteria. They are oxidizing agent. As example, iodine for skin and chlorine for water. Salts of heavy metals as Mercurochrome for wounds. They are antiseptics that cause denaturation of the protein of the bacteria and can be used safely as a disinfectant on a living tissue. The final disinfectant used here is Cidex 2%. Cidex 2% concentration, and its scientific name is glutaraldehyde. Glutaraldehyde. This preparation is very important, very important. It's considered sterilant. That means it causes sterilization. That means it causes high level of disinfection. Why it is very important? Because Cidex 2%, when the instruments come in contact with all the instruments that come in contact with the mucous membrane and abrade the skin used in the hospitals, must be sterilized. All the instruments, all the tubes of the respiratory systems and dialysis systems must be changed from one patient to another. All this instrument is disposable and must be changed from one patient to another in endoscopes and others. Okay, but due to our limited resources in our hospital, we can't make this option, but we can sterilize these articles and reuse it safely. Meticulous cleaning of these articles with detergent and then soaking in Cidex 10% for 20 minutes will give high level disinfection or complete sterilization. So it is life-saving in some conditions. Just soaking in the Cidex 2% for 20 minutes will give sterilization of these articles and can be used safely in contact with the mucous membranes or in abrasive skin from patient to another. Okay, so Cidex 2% or glutaraldehyde is very important to know it, and very important to understand that it gives sterilization or high level of disinfection and can be used for the endoscopes, sterilization of endoscopes, thermometers, and all the instruments and the tubes in the respirators and dialysis system. Finally, we will talk about the gaseous disinfectants. the gaseous disinfectant, and the most important example is the ethylene oxide gas. Ethylene oxide gas. This is the lethal agent used to kill the microbes and its spores. That means it is sterilant. Sterilant, that means it causes sterilization. So ethylene oxide gas is a chemical disinfectant, but it is sterilant. It is alkylating agent that replaces the hydrogen in the reactive groups of protein in the bacteria by a hydroxyl ethyl group or any other group that make it inactive. It is used for articles liable to damage by heat. It is used for articles liable to damage by heat, as plastic, any plastic syringe or any plastic pharmaceutical products, rubber articles, and catgut. This is used in the surgical sutures. Catgut, which is surgical sutures. All these products is sterilized by ethylene oxide gas, and in the Asset University hospitals we have 2 ethylene oxide gas apparatus. But an important point, you should know that the cycle of the ethylene oxide gas apparatus is nearly 18 hours. The cycle of the apparatus nearly 18 hours. That means when I enter, the equipment required to be sterilized in the ethylene oxide gas, and I begin to make it on, the apparatus on, it will take 18 hours in order to obtain the product sterile. The sterilization is only for the first 6 hours. The first 6 hours is sufficient to achieve complete sterilization. But the rest of time is required to remove any traces of the gas present inside or on the articles, to free the articles from any residual effect of the ethylene oxide gas. We require additional 20 hours for the ethylene oxide gas apparatus. It is very important to know. And so when we are talking about side effects, I said that it is life-saving in some conditions, because soaking in Sidex 10%, 2% for 20 minutes only will give sterilization. So if it is a operation that is rapidly required, the Sidex is life-saving. Ethylene oxide gas requires much more longer time. The efficacy of ethylene oxide gas must be also monitored from time to another, and the monitoring system here is biological indicator containing the spores of organisms that is very resistant to the ethylene oxide gas called Bacillus atrophius. The final point in the ethylene oxide gas, you should know that it is explosive gas and so must be mixtured with 10% carbon dioxide inside the ethylene oxide gas apparatus chamber. The formaldehyde gas also is another gas, but it's only a disinfectant and not reached to the sterilization level, and used for articles also, instruments, shoes, hairbrushes, boxes, and other non-living articles. Thank you.